Thanks for joining us. And I'm really happy today to have the opportunity to introduce Dr. Alyssa Deming as our speaker. And we share that some connections because we both spent time at the Marine Mammal Center as well as several other places. So I'm going to just read an excellent bio that she provided, and so that you can know a little bit more about her background and experience. So Dr. Alyssa Deming is the Vice President of Conservation Medicine and Science at the Pacific Marine Mammal Center, which is in Laguna Beach, California. And just an aside as well, that 2021 is the 50th anniversary of the Pacific Marine Mammal Center, so a lot to celebrate there. As the Vice President of Conservation Medicine and Science, she's responsible for the medical care of dolphins, pinnipeds, which include seals and sea lions, and whales that strand in Orange County, California. So that's the Los Angeles, Laguna Beach, Long Beach area. Dr. Deming received her doctorate in veterinary medicine and PhD in comparative diagnostics and population medicine from the University of Florida. Her principal research interest is the study of disease patterns in marine mammal populations to help better understand the impacts of human and environmental factors on ecosystem health. Her primary research efforts are focused on a commonly diagnosed cancer in wild California sea lions, which we're going to hear more about today. And she is the chair of the Sea Lion Cancer Commission. She has extensive veterinary medicine background, having worked as a clinical veterinarian at the Marine Mammal Center in Sausalito, SeaWorld San Diego, Dauphin Island Sea Lab, and the U.S. Navy Marine Mammal Program. She has provided veterinary care for a variety of aquatic species in rehabilitation, managed care, and field settings. And her background includes extensive experience in remote sedation and field disentanglements of pinnipeds and cetaceans, as well as performing necropsies, postmortem exams on pinnipeds, dolphins, whales, manatees, and sea turtles. So I'm excited to hear an update on your cancer research. So take it away.
Awesome. Thank you, Dr. Nutter. I am really excited to be here today to talk to you guys about what is my favorite topic. If I've cornered any of you at conferences or workshops, probably talk to you about viruses or herpes and cancer and contaminants. So this is my favorite topic, the area that I did my PhD work in, and continuing to do a lot of work with the Cancer Consortium, as well as a variety of rehabilitation facilities and universities. So I'll give my little pitch at the end that if you guys are interested in potentially collaborating on some kind of cancer research, we do have a Sea Lion Cancer Consortium that has a great archive full of samples that Dr. Francis Gullin started collecting about 25 years ago at the Marine Mammal Center up in Sausalito. And now a variety of stranding networks along the West Coast have started collecting samples to help contribute to that archive. So a lot of the stuff we're going to talk about towards the end of this presentation today are from samples collected in that archive, and we have lots of samples remaining for future work. So I'll give you guys a contact at the end of the talk to reach out if you guys are interested in potentially coming up with some brilliant ideas after hearing this talk today. So like Dr. Nutter said, I am at the Pacific Marine Mammal Center, which is in Orange County, California, beautiful Southern California. The southern bite of California with the Channel Islands right off our coast is kind of where we're going to be focusing our talk today. But sea lions generally use the range along the entire West Coast. And there are stranding networks similar to ours, rehabilitation facilities that do different degrees of research, but our primary goal is rehabilitation of sick and injured marine mammals, as well as obviously education and research as part of just our foundation and our mantra here. It's important to educate the public about marine mammals so we can help take care of them and our environment. So before I get too deep into it, I just want to acknowledge that I am lucky enough to have one of the coolest jobs. I love working with marine mammals, and more importantly, I love working with the people that are attracted to this field. So I want to give a shout out. It is Vet Tech Appreciation Week. That's our Vet Tech Jen in the upper right corner with the red hair, viciously taking notes on one of our patients, Muffin, an elephant seal under anesthesia. We also have veterinary assistants, students, and a whole team of animal care staff that are integral in providing the care for our patients while they're in the hospital. And then on top of that, we have a lot of volunteers that without which we couldn't do the amount of work that we do. We are a small nonprofit, so we run off of donations. So we couldn't afford the staff that's necessary to do the level of animal care or the level of research that we do if it wasn't for students, volunteers, docents, and collaborators kind of working with us on these projects. So at the Pacific Marine Mammal Center, the variety of the patients we do see are pinnipeds. So those are seals and sea lions. And we see a variety of those types of like otarids and phocids. So we'll see California sea lions, that's our #1 patient, and of all sizes, mostly little babies, but we also do get subadults and adults for sure through here. And then we see elephant seals, which are the 3 big ones you see across the top of the screen, as well as harbor seals, Guadalupe fur seals, northern fur seals, and then dolphins and entangled whales. We will go out and do responses on the water. We have a big entangled whale team that works with NOAA to help go out into the water to disentangle any whales that need our assistance off our coast as well. So today we're going to kind of talk about what opportunities there are to do research with our patients when they come through the hospital. So something to consider with the work that we do here is our primary goal is animal care. So we want to rehabilitate these animals and get them released out into the wild. But during their stay here, or if they need to be euthanized or pass away, we can do necropsies on them and collect samples from them, or bank archive samples from diagnostic testing that we run while they're in the hospital. And that allows us to do a variety of the research projects we do and that we're going to talk about here today. So like I mentioned, it's not just the animals that we bring to the hospital. We also, a lot of times our patients are quite large, so we need to go to them. So we do kind of take the show on the road and go into the field whenever necessary. So one of the things that we're starting to get ramped up and get a little bit better at the Pacific Marine Mammal Center is doing disentanglements of both pinnipeds and whales, as I've mentioned. And so we see a lot of fisheries interactions, unfortunately, with pinnipeds that get entangled in fishing gear, like that upper left-hand picture. So we go out and do remote sedation in order to get our hands on these animals, either disentangle them in the field or bring them back to the hospital if they need more surgical intervention. And then get them back out into the wild. That's one of my favorite things to do. And then in addition to that, we also support field work. So I've been lucky enough to go to San Miguel Island. That is the primary rookery for California sea lions, northern fur seals, and some elephant seals. And we're going to be focusing a lot on the population of animals there in today's talk. And I'll kind of explain a little bit more about their life history and the important role of those islands in the sea lions' life history in general, and also how those islands may be playing a role in the development of this common cancer we see in California sea lions. So I just kind of want to acknowledge that today I'm going to be doing a lot of talking. I'm not going to focus too hard on the research. I want to tell the story of California sea lion cancer, kind of let you guys know the things that we kind of understand about it now that we have this general foundational knowledge, and then kind of hear about potential ideas that you guys may have during the question and answer session about what additional research we can do in the future, and any ideas if you guys are good in endocrinology, contaminants, things like that, that you may think could help contribute to our body of knowledge about environmental health, individual animal health, and also using these sea lions as a model for understanding how virally induced cancers and contaminants can cause disease in mammal species in general. So that's kind of the pitch for this. I will say I am very lucky, as we were discussing in the beginning, to have some pretty cute patients. It's pretty easy to get people to be interested in California sea lions. And when your elevator pitch is that they have a sexually transmitted herpes virus that causes genital cancer, it's actually a great platform to start getting conversations started. I should warn you that right now you probably think this picture of this baby sea lion pup nursing on his mom is adorable. By the end of the talk, you may have a little bit of reservations about these nursing sea lion pups and some concerns about some infectious diseases moms have. So that's my little disclaimer that I may ruin this cute fuzzy picture for you in the near future with the reality of what's going on in their environment and how it's impacting individual animals and the population as a whole. So as Dr. Nutter said in the beginning, my research interest is really trying to use marine mammals as a sentinel for environmental health. So Sea lions in particular are great at this because they're very coastally associated. So they live along our coast. They eat similar food and prey items that we would potentially eat, or that the fish that we eat, like tuna, eats the same prey items like the sardines and anchovies and things like that, that the sea lions feed off of. So they're a great model for contaminant biomagnification and understanding. They're kind of like the sponges of the sea. They'll soak up any contaminants that are within their environment. So by testing their blubber, we can kind of get a snapshot into the things that they've been exposed to throughout their life. That's also an unfortunate thing because these sea lions are mammals, and I like to remind people of that. So they give live birth, they lactate, they have hair, they're warm-blooded. Their anatomy, physiology, immune system, infectious diseases are very, very closely related to humans and other mammals like cats and dogs. So although we like to think we're different than animals as people, our anatomy and physiology is exactly the same. We are actually within the animal kingdom. So that's why they make a really good model for understanding how disease processes develop and how the environment and infectious disease in their population may mimic what we're seeing in human populations. So the main focus that we're going to be talking about is an environmental contaminant called DDT, as well as PCBs that are kind of in the area, and we'll explain why shortly, that sea lions kind of give birth and raise their young, and the females feed in, and how that impacts the infectious diseases that they're exposed to and the host's immune system, and how these multiple factors play into the development of a disease process. So there are a lot of inherent challenges working with wildlife kind of in situ out in the environment, as opposed to working in a lab with lab animals. Obviously, with lab animals, you know the animal's history, you can control a lot of parameters, and then you can just tweak small things in your experiments in order to tell which things are having impacts at what concentrations. We unfortunately don't have that option when it comes to marine mammals. And we're kind of doing a global experiment, whether people realize it or not, by putting things into our environment and then having animals live in those things. Natural processes are going to happen. So rehabilitation centers like the Marine Mammal Center, the Pacific Marine Mammal Center, and others along the California coast have tried to figure out how we can utilize the patients that come through our hospital as a sounding board for the problems that exist in their environment. So there's a lot of research that's done in this area, and it's definitely taken a village. So these are just literally a handful of publications on sea lion cancer. In the mid-1990s, Dr. Francis Gullin started working at the Marine Mammal Center in Sausalito, California, which is a very large rehabilitation facility that covers about 700 miles of California coast. When she started there, she was realizing that a lot of the sea lions, an unusual number of the sea lions, were presenting with the same type of genital cancer in about anywhere from 15 to 25% of the adult animals that were coming across the necropsy table. So Dr. Gullin had the foresight to know she needed to look further into this, and in order to understand what was going on and why this wild species was having such a high rate of cancer, she was going to need to collect samples in a systematic way. So over 25 years, she set up a cancer control protocol that allowed her to collect various tissues from California sea lions that had cancer, that either died or were euthanized because of a grave prognosis or end-stage disease, as well as California sea lions that died of other things, most of which, for the control cases that we'll be talking about today, were associated with trauma, shark bites, gunshots, fishing interactions, as well as domoic acid intoxication. So by having freezers full of samples from animals with and without cancer, that allowed her to set up this massive cancer control study that was just recently published earlier this year in Frontiers. That's the bottom right-hand citation on your screen. We'll be talking about that just briefly towards the end of the talk. But I think part of her brilliance is the ability to break up big questions into smaller pieces and make progress in understanding the disease over those 25 years while simultaneously collecting tissues for a larger study. So initially it was identified that there was this common metastatic cancer seen across adult California sea lions. It looked like it started in the genital tract of them, but then it would aggressively metastasize and practically spread everywhere. And that would end up causing the animal to be in end stage cancer and then either strand on the beach or die naturally. And then we would necropsy them and collect these samples for the archive that is housed at the Marine Mammal Center through the Sea Lion Cancer Consortium. Then there's been a lot of work on things like genetics. Are sea lions predisposed genetically to get cancer? That's definitely a thing. People think of the BRCA gene in humans as a good example. So there's been some work looking at genetics to see if they were predisposed. And we're starting to think maybe not, but there was initially some evidence that there might be some MHC molecule predisposition in some of the animals that looked a little bit inbred because of their breeding strategy. And then my PhD was associated with the two bottom papers. So Francis tasked me during my PhD to basically prove that a herpes virus that's been identified from the genital tract is actually oncogenic. So there's always that big question when you run diagnostic tests, is it correlative or is it causative when you pick up a bacteria during a culture or a virus from a swab? Short of being able to prove Kutch's postulate by infecting a bunch of sea lions with herpes viruses, giving them a bunch of contaminants and seeing if they develop cancer, we had to figure out a way molecularly to try and determine if more definitively if this herpes role was just along for the ride or if it was actually inducing the disease. So we'll be talking about a little bit of that today. And then this is going to be my pitch for the Sea Lion Cancer Consortium. This is our most recent meeting, which sadly enough, I think was in 2018 or 19, but we try and meet at least every 2 years and then remotely a couple times a year. And this just gets all the researchers that are doing sea lion cancer in a room together to kind of go over the findings, make sure we're not duplicating efforts, dividing the wealth among collaborators to make sure we have samples to go around and to have some kind of organized movement towards an end game to help better understand this disease and utilize these animals the best way possible for understanding environmental health, as well as using them as a model for studying metastatic cancer and virally induced cancers. So before we get too deep into infectious disease and contaminants, I want to kind of tell you a little bit about sea lions and their life history. So this is one of the most beautiful places on Earth, San Miguel Island. And on the waterline, you can see all those little dots. And all those little dots are northern fur seals, California sea lions, and a sprinkling of some elephant seals. And San Miguel Island and just the Channel Islands off the southern coast of California are where California sea lions go to breed in the summer, have their babies in the summer, and then the boy sea lions kind of migrate up north during the fall, winter, and early spring months. And the female sea lions are tied much closer to the island because they have a dependent nursing pup. So this is just showing you from NOAA's stock assessment, the different stocks of California sea lions on the West Coast and where their rookeries are. So that line dividing the North and the South is the border for Mexico and the US. And there are 3 rookeries in Mexico that are kind of identified to be somewhat genetically unique. And then there's one in the Channel Island area off of Southern California where California sea lions give birth. And the high rate of cancer has only really been documented in the US stock of sea lions that are giving birth on the Channel Islands. There's a great stranding network in the US. There's also a good stranding network in Mexico, but the efforts for rehabilitation, necropsy, and research in the US is pretty heavy. Whereas in Mexico, those islands are a little bit more remote. It's hard to do necropsies on fresh animals. So although they don't see as much cancer and very little is actually reported in that population, the effort is a little bit less. So we do have to take that into consideration and maybe try and figure out if we can look a little bit harder to see if the cancer is actually there and we're just not picking it up, or if it is a unique epidemiology or disease that we see in the Channel Island animals. Oh, other way. So this is just a close-up of where those rookeries are. The Channel Islands consist of maybe a half a dozen islands kind of between Santa Barbara and San Diego. And this area is just one of the most nutrient-rich areas we get during El Niños or during La Niñas, this upwelling of cool water and nutrients, which brings in a lot of prey items. So there's whales and large pods of dolphins. And one of the reasons why California sea lions and northern fur seals chose these islands to have their babies on is because there's easy, food right off of the coast. So they have these dependent pups, the females. California sea lions nurse their young for about 9 months to a year, and they have a baby almost every year, if not every year. So they're very tied to these Channel Islands when it comes to where they're hunting. And then the males kind of move up north, like I said. So this is showing you some tracking of 7 female California sea lions that just kind of show where their foraging behavior is. And this was a study done in the Costa Lab from 2003 to 2008. So it was a little while ago, but it just goes to show they do migrate up and down the coast to feed. But they only go on trips for about 3 to 7 days, and it depends on where the fish are, and that's where they typically go. So they have different foraging patterns during El Nino and La Nina years, but the vast majority of the California sea lions are nursing right off of the Southern California coast, or not nursing, well, they are nursing, but feeding, and then nursing their young right off the Southern California coast in the waters right around the Channel Islands for the most part. So the reason why I'm pointing that out is because this is a close-up video or picture of Catalina Island, which is one of the more southern Channel Islands, right off the coast of Orange County, basically, and LA. You see the Palos Verdes Peninsula right up there. And there's been a long known point source of DDT being discharged into the water right off of LA coast. So for those of you that aren't familiar, DDT is an insecticide that we used to use in the 40s. It was, I think, discovered in the 40s and used pretty heavily in the 50s, 60s, and then started being pulled back a little bit in the 70s and 80s because they started realizing it had an association with causing cancer. There's a lot of articles about DDT and the impacts on humans that have been exposed, and it can be a transgenerational impact. So if your grandmother was exposed to DDT in the 60s, your risk of getting breast cancer actually is 33% higher than somebody who didn't have a grandmother exposed to DDT. So this has a very real human health and public health impact. And so I encourage you to dive into the literature and learn a little bit more about the impacts on people. But today we're going to focus on the impacts on sea lions and how they may be sounding alarm bells for what this DDT could mean for all of us, not just the sea lions in the environment and not just people, but just marine organisms in general. And then more recently, researchers from UC Santa Barbara had an underwater vehicle kind of just looking around for some weird pings they got on their sonar. And they picked up a dumping ground of DDT barrels, the size of Manhattan. I think they're saying like over 50,000 barrels probably of DDT that was illegally dumped. So in the 50s and 60s, we knew there was a point source of people just dumping DDT down the drain because there was a big factory right there on that peninsula. But after they were being reined in by the government, and there was litigation and lawsuit for that point source, apparently there was also dumping that was happening illegally of these barrels kind of all around the Channel Islands, and most recently discovered right off the Catalina coast. So the problem with DDT is it's something called a legacy contaminant, and I'll show you what these bottles or barrels look like. This is kind of an image from the ROV that was underwater that captured a barrel of leaking DDT. And so there's a lot of measurements being done to determine how much is in the sediment. This goes to ask a bigger question of what do we do about all the DDT barrels that are down there, and that's actually out of my area of expertise. It's going to take an engineering feat to figure out how to deal with these. Corroded barrels filled with toxins at the bottom of our ocean under high pressure. What does it mean if we bring them up? Is the cleanup going to make it worse than better? So all of those questions I don't have answers to. I'll just preemptively warn you. But what I can tell you is this DDT not only is persistent because of these barrels, but also persistent because of sea lions' life history. So these sea lion moms are feeding all around where these DDT barrels are being dumped. And there has been research looking at DDTs and PCB levels in sea lions, particularly those with cancer. And we have found that sea lions with cancer, or sea lions are more likely to have cancer if they have high levels of DDT. The problem with DDT is it's a legacy contaminant. So not only does it last in the environment and in the sediment for hundreds of years, if not longer, it bioaccumulates up the food chain. And then these nursing moms have DDT in their blubber. And when they have their pup, they mobilize that blubber to make their milk, and the pup drinks the milk, which is latent with the DDT contaminant. So that's why it's transgenerational and can be passed down from one generation to another. These moms are preloading their pups with DDT before they even start eating fish, just from the milk that they're drinking. So there's a lot of research that needs to be done to better understand how this DDT is impacting these sea lions. Is it causing immune suppression? How is it predisposing them to cancer? DDT and PCBs, which is another type of polyaromatic or legacy contaminant, long-term contaminant that is around these islands associated with a lot of bombing practice that was done in the 60s and 70s. We need to do a lot more work to better understand how it's impacting these animals' long-term health. And so we're just getting to the answer, or getting to ask those questions now that we've effectively determined that DDT is one of the components that's contributing to this high rate of cancer. And when we say high rate of cancer in California sea lions, we're talking really high. They have one of the highest rates of cancer of any wild animal out there. And in the adult animals that we necropsy, about 20 to 30% of those animals have aggressive end-stage metastatic cancer that starts in their genital tract. So it's not just the DDT and the contaminants. If that was the case, probably all of the sea lions would be presenting with cancer, or at least a lot more of them. But like we had talked about in the beginning, there is an infectious disease component. So this is a beautiful image that somebody made for a cover story about sea lion cancer. And it's very profound to me because it kind of reminds you, if you can see those little shadows of giant virus particles, for those of you, most people now can recognize virus particles, unfortunately. The sea lions are, swimming in the sea of virus, just like any other species would be. So sea lions have unique herpes viruses. They actually have unique coronaviruses, believe it or not. So every mammal species has their own groups of viruses that impact their health. And researchers determined that there was a high rate of herpes in the genital swabs and tumor tissues in these California sea lions. And whether or not you realize that herpes is kind of everywhere, there's 8 herpes viruses that infect humans, 2 of which are known to be associated with cancer. So we do know herpes viruses can be oncogenic. So we wanted to do some further work to understand, is this DDT causing immune suppression or affecting estrogen or progesterone receptors in the genital tracts in a way that could be kind of promoting this herpes virus to go from a latent state to a more lytic or oncogenic state in an infected patient. So just so you guys can see what some of this cancer looks like and get a better understanding of what it takes to get the samples that we do this research on, this is an adult female California sea lion that's showing classic paresis or paralysis of the hind end. She's got swollen hind flippers that have pitting edema, and she's in poor body condition. So when California sea lions present like this is almost pathognomonic for a urogenital carcinoma. And we'll show you some necropsy pictures shortly to explain why they walk like that. But all sea lions at the Pacific Marine Mammal Center and at the Marine Mammal Center up in Sausalito will be put under general anesthesia for their admission exam. And they get like the best doctor's appointment ever right when they walk through the door. They get blood work, physical exams, ultrasounds, radiographs. We do lots of swabs to kind of determine what the cause of their stranding is. So this is my warning to everybody. We're going to start getting into some gory pictures. It wouldn't be a veterinary presentation if there weren't some necropsy pictures, and it's quite profound to see what they look like. So I know a lot of you are probably zooming from home, maybe have some small kids in the room and don't want to see pictures of these things. So this is the time to kind of turn your camera or your computer away from them so they don't have to see stuff that you don't want them to see. So some of the classic signs that these animals present with are these swollen vulvas or prolapsed penises. And that's practically pathognomonic for urogenital carcinoma. And they can have this abnormal tail position and also have urinary incontinence, as well as urinary obstructions associated with a mass effect from the intra-abdominal metastatic lesions, or from the primary mass on the penis can result in a male's penis being prolapsed, and then obstructing the ureter, preventing him from being able to urinate. So that's kind of a classic sign that we see in some of our patients. Like I said, they all get put under anesthesia. We collect lots of blood from them. All of that gets banked and put into the archive for future work, in addition to working, obviously, them up diagnostically to determine why they stranded. And then on ultrasound, this is evidence that I should mention marine mammals have weird kidneys. They're reniculated, kind of like cows. So although these kidneys are abnormal, that those round reniculi are a normal thing for marine mammal kidneys. But that giant tube underneath is actually a ureter that's distended with urine. And that's one of the classic signs that we see as clinicians to determine that these animals have urogenital carcinoma, is urinary obstructions, ureter obstructions, or hydronephrosis, which is a backup of urine in the kidneys. So the reason why that happens is the primary mass starts in the genitalia. So on the left is a plaque lesion on the penis of a male California sea lion. On the right is a plaque-like lesion on the cervix of a female California sea lion. And those are kind of early stage, quite subtle lesions, but those animals could already have very aggressive metastatic disease. And then this just shows you how aggressive those primary lesions can look. This is a prolapsed penis in a California sea lion that had a super distended bladder because he couldn't urinate anymore. And then on the right is a uterus, ovaries, and kind of repro tract spliced open that's showing you a large mass in the uterus and then a pretty large mass down there in the cervix of urogenital carcinoma. Unfortunately, it doesn't just stay locally. So this is a California sea lion opened up. His head is towards the left. His genital tract is towards the right. And all of those white dots you see on the liver are metastatic lesions. And this is a quite classic presentation where we see carcinomatosis and metastatic lesions to the lung, liver, lymph nodes, spleen, kind of goes everywhere. And then this just shows how it can preferentially spread to the lungs in some patients and just be riddled with metastatic lesions in the lungs. And then the liver can be clean, and then vice versa, or it can just be everywhere. So this is a close-up of the kidneys. I'm not sure if you guys will be able to see my pointer, but these are big kidneys, and right in the middle of your screen are just giant sublumbar lymph nodes. And it's those metastatic lesions that are obstructing the ureters, causing that urine back up into the kidney, allowing us to see that hydroureter and hydronephrosis on ultrasound. And then this is what it looks like when you take those out. So this sea lion conveniently got metastatic tumors on the left-hand side of its urogenital tract, but not on the right. So you can kind of see what a normal kidney would look like, a normal ovary and a normal uterus. And then you see this giant metastatic lesion wrapping around that ureter, causing hydronephrosis in that other kidney. And it can be quite extensive. So this kind of almost looks like a cauliflower farm in their abdomen when you open them up. And all of these white firm masses right in the middle of this screen are metastatic lesions, as well as all those mets kind of sprinkling the serosal surface of the kidneys. So I just kind of wanted everyone to see what that looked like so you can get an idea of how aggressive and terrible this disease is and why there's no way that we could actually when these animals present in end-stage disease, end up treating them. But as a clinical veterinarian, I would get really frustrated with these cancer cases. And that's why I went back and got my PhD, to start seeing if we could utilize these animals to help us better understand how viruses can cause disease. So we're just going to go over it very briefly. I encourage you to read some of the publications that recently came out if you want to dive a little bit deeper in this. But for time's sake, I'm just going to say that herpes viruses are everywhere. By the time you're 24 years old, about 100% of people are infected with at least one type of herpes virus. And of those herpes viruses, like I had mentioned before, 2 of the 8 are oncogenic or can cause cancer in some people infected, but not all people infected. So that's why I refer to herpes as kind of like the barometer of the immune system. So a good example in people is Kaposi sarcoma in AIDS patients. So when HIV/AIDS wipes out the entire immune system and your immune system can't keep herpes virus suppressed, that's when Kaposi sarcoma comes out in those AIDS patients and develops cancer. And we're wondering if we can use this otorine herpes virus 1, which is the cancer, the herpes in the sea lion cancer, as a barometer for the immune function of the sea lions to help determine if the DDT is actually causing immune suppression, which is allowing this herpes virus to gain a foothold and cause such a high prevalence in cancer. And not only is herpes something common in both humans and sea lions, but so is cancer. So if you're a woman, you have a 1 in 3 chance of developing cancer in your lifetime. If you're a man, a 1 in 2 chance. And not a lot of people realize that 20% of cancers are associated with infectious disease. So if we can figure out to either how to prevent infectious disease or keep it from transitioning into an oncogenic state in the sea lions, we could potentially use those kind of preventative mechanisms to keep almost 20% of cancers in people from developing. So this really does have a big One Health component and a lot of potential for better understanding these multifactorial disease processes. So this is just a quick look at one of the papers that recently came out, how we proved that the otorine herpes virus 1 was actually causative and not correlative in urogenital carcinoma in sea lions. And the first thing we saw that by doing qPCR, which is a quantitative method of measuring viral load, the sea lions with urogenital carcinoma had close to 1 million to 10 million virons per nanogram of DNA. Whereas animals that were infected with the herpes virus but did not have cancer only had less than 10,000 virons, so exponentially less viral load. And that's probably because those sea lions' immune systems are functioning appropriately, keeping that virus suppressed and behaving itself, whereas in the urogenital carcinoma, that virus is being heavily expressed. So that was our first clue that in these cancer cases, this virus may be playing a significant role. And I'm going to skip over that slide. So this is a histosection of a cervix of California sea lion. And you can see on this histosection that the epithelial layer of the cervix is normal. Cells are supposed to follow rules. They're supposed to die when they're supposed to. They're supposed to stay in the appropriate neighborhood that they're supposed to. So they're not supposed to migrate to different places. And they're supposed to divide when they're told to, but not otherwise. If you look on the right-hand side where this tissue is diving down deep into the underlayer of the epithelium, that's carcinoma in situ. So that's cancer starting. So we wanted to know if this animal is infected with herpes, is the herpes being actively expressed in that normal tissue as much as it is in that diving down cancer tissue? Or is the herpes more activated in the cancer tissue and not in the normal tissue? So we made something called RNA scope probes. And these are probes that are made of RNA, messenger RNA. So it's specifically looking for viral expression. And we targeted them to some potential oncogenes, suspect oncogenes in the herpes virus after we sequenced it. And you can see on that upper right-hand corner, which is the RNA scope probes looking at the virus, that the probes only bound very heavily to the cancer tissue, but not to that normal cervical epithelium. So that is kind of a slam dunk, as good as you're going to get to prove that this herpes virus is actively being expressed in this cancer tissue, not actively being expressed in healthy tissue. So is most likely driving the disease progress and the cellular transformation onto the oncogenic pathway of developing your urogenital carcinoma. And then the next study, oh, there should be something that says control there, but the next study recently published this year was that big 25-year cancer control study that Francis Gullin really worked on spearheading for 2 decades. And this was a great epidemiological feat that Elsa Hall helped with, and she did odds ratios and looked at different parameters and determined the likelihood of developing cancer if you had certain things. So we looked at genetics, we looked at contaminant loads, specifically DDT and PCVs, and we looked at herpes infections. And although this may if you're a mouse researcher, this may seem like a small sample size, we had 179 cancer sea lions that we collected from a 20-year period and 215 control samples. And that's actually a massive number of animals, especially marine mammals, to include in a study like this. So it was a very powerful study. And we did find that the sea lions that had cancer were more likely to be infected with the herpes virus that was required for them to develop cancer in the predictive model. And then the animals that did have cancer also had very high levels of DDT and PCV that were significantly higher in the cancer animals than the sea lions that didn't have cancer. So now I bring you back to this cute picture of this pup nursing on its mom. And hopefully you're seeing what I see now as a virologist and someone who studies cancer, that she probably got herpes, sexually transmitted herpes, when she mated with a male. And if her immune system is acting right, or her receptors are right, or she's not exposed to DDT, she likely will never develop urogenital carcinoma. Just like somebody infected with Kaposi sarcoma herpes virus, if they don't get HIV, or if their immune system is functioning appropriately, they won't develop Kaposi sarcoma in their lifetime. So by utilizing these sea lions, we can help better understand what's triggering this virus to start causing cancer, and that will help us prevent it from happening in people potentially in the future. And then in addition to that, the sea lion pup is getting its first taste of DDT. So this sea lion was likely feeding around the Channel Islands, which is where all those DDT barrels are spilt, and that's bioaccumulating up in the environment. She likely has DDT and PCV accumulated in her blubber, and when she's mobilizing that blubber and nursing her sweet little baby, it is getting its first exposure to DDT. So that is why these animals just make an excellent comparative model for understanding the complicated webbing of what happens with infectious disease, environmental components, and it's not just contaminants. It can be stress associated with lack of nutrition. It can be stress associated with climate change and needing to change migratory patterns. It could just be generalized reproductive stress. These sea lions are reproductive machines. They have a very interesting delayed implantation, a lot of hormone cycling that happens. And the beauty of studying sea lions is that they have synchronized birth and synchronized estrus. So we can actually tell where their hormones should be at different points of the year. So in an animal that we want to study genital cancer in, that's very useful, especially since we can't control a lot of these parameters in the lab. So this is just my little pitch of saying that California sea lions can be used as a translational cancer research model, which is something that NIH is already starting to push with companion animals. But we are recommending that you can actually put a sea lion in that place as well, and we can utilize the information we're getting from our patients that come through our hospital to better understand how cancer develops in a naturally occurring disease model, how viruses are actually playing a role in the development of cancer, and to better understand how cancer spreads through the body, or metastatic disease, and the processes associated with genes being turned on and turned off for cells to be able to mobilize and settle down in distant sites, causing end-stage cancer in people. And then keep in mind, if you're good at contaminants, if you know a lot about immune systems or the endocrine system and hormones, I would love to talk to you a little bit more about anything you think you can do with some of these samples. And I do think in the future, we could potentially look at some treatment options, because we do occasionally see this in California sea lions at aquariums, but they typically don't develop the cancer, the urogenital carcinoma, until later in life. And that is because it's more likely that when they're not exposed to DDT in these aquariums, but as your immune system wanes later in life, that may be giving the opportunity for the herpes virus to gain a foothold and cause cancer in those geriatric managed care animals. So there could be an option of studying them in managed care long-term. And then this is just all the names of the California sea lions that participated in my PhD study. So I want to give them a shout out. Unfortunately, all of them either died or were humanely euthanized. But me as a clinician, being able to do research like this and kind of let them live on to inform us more about environmental health, cancer, those kind of things, is kind of the silver lining, and keeps us going when we're doing this challenging work. So this is where you can go if you're interested in learning more about the Sea Lion Cancer Consortium. On the very bottom of the page, there's kind of like a contact us link where you can put in your email and kind of what you're interested in the little comment section. And I will get back to you and see if we can plug you in with some samples or some of our experts that are doing work in your field of interest with California sea lions. And I think that is all I've got for you, and we should have a little bit of time for questions.
Thank you so much. It is perfect timing. We do have a question. We have, do you think that multiparous California sea lions are at a less risk of urogenital carcinoma because they are essentially decreasing their bioaccumulated DDT/PCBs with lactation?
Yeah, that's a really good question. The male sea lions for sure have significantly higher levels of contaminants than the females because they don't have that yearly offloading of contaminants to their lactating baby. So females do have lower contaminant burdens, and their first offloading, so the first pup that they have gets the highest levels, and it does go down throughout after that to their next pups. But we don't really know the role that DDT plays and what the critical threshold is for it to be impacting whatever system it is to predispose these animals to developing cancer. So we actually think it may be playing a role either in utero or as an animal sexually matures with the kind of estrogen and progesterone receptors on the reproductive tract. And that's been shown to happen in humans. So that's an area of research that we're really interested in pursuing, is to better understand the receptor patterns in sea lions exposed to DDT early in life versus a control sample, wherever we can find them, maybe like, I don't know, the Galapagos sea lions that maybe don't have that DDT exposure, to see if it's impacting actually their receptors well before, you know, they're developing the cancer, if that makes sense.
Let's see, does the DDT content in the milk result in higher mortality rate in the seal pups?
I don't know if that question has been asked scientifically, and it would be a little bit challenging. I don't think that they get, this is just me speculating, I don't think they get a big enough hit to kill them straight away. But, you know, they do manage to sequester it into their blubber. So it's usually not very bioavailable until they start burning that blubber layer. And then you can kind of go into how sea lions are masters of starvation, and these pups have these waxing and waning, like weight gains and weight losses. So maybe it is more mobilized, but that's definitely an area where more research needs to be done. And now that we've kind of realized that there's more DDT out there than we knew about, and that it's having a major impact in this population's health, I think there will be more effort in doing that kind of research to help tease out the impacts that it's having on these animals, and then potentially, you know, marine animals and people across the board.
Okay, thank you. Can the herpes virus in California sea lions be passed along while nursing?
That's a good question too. We don't think so. It would make sense when they're being born, like through the genital canal, that they're being contact exposed to it. But very rarely do we see herpes positive genital swabs in pups, yearlings, or juvenile animals. Every once in a while we'll get one, but it's very rare. Whereas when we start taking genital swabs in reproductively active animals, even before we see the cancer, anywhere from 30 to 50% of those animals have herpes viruses, or have specifically the odorine herpes virus 1 in their genital tract. So we really do think it is a sexually transmitted disease, but it doesn't mean that it can't, on rare occasion, be transmitted to the pups.
Okay. Let's see. Has there been any update on the removal of the DDT barrels in the bottom of the ocean?
Yeah, I don't know what the right answer to that is. Well, I know that very smart engineers and are trying to figure out ways to remediate that situation. You can get into a lot of trouble digging up sediment when contaminants have already settled down into that sediment. You may end up mobilizing more of it than would have ever been mobilized if you just left it alone. There's some talk about trying to just bury the barrels, just putting a bunch more stuff on top of it to entrap it under the seafloor. But it is going to be an incredibly expensive and basically a very challenging engineering feat to figure out how to get these corroded, leaking barrels contained and brought to the surface to bring them out of the ocean. So I'm hoping that someone comes up with a brilliant idea to figure out a way to deal with this, but I haven't been able to come up with any.
It is mind-boggling.
Yeah.
We have a few more questions, but we also need to wrap up so students have time to get to class and such. So the remaining questions we have recorded, and we can get to after this by email, and I can follow up with anyone that way. But I wanted to also give you a chance to wrap up and say any last words that you'd like to say.
Yeah, I just want to say thank you guys for having me. I think it's a great platform that you guys are doing this at Tufts, and I'm hoping that some of our volunteers and staff are going to start sitting in on your talks. So I appreciate you guys having me. I think this is a major issue, and for those of you in vet school or those of you that are graduate students and interested in doing research, we do have a formal veterinary externship where you can shadow and do clinical stuff here at the Pacific Marine Mammal Center. And we do lots of collaborations with primary investigators that need samples. And we also let students come here and do work to help collect their samples. So if it's something you're interested in, please reach out, and we would be happy to figure out how we can get you out here to help us. And I think just my final pitch is for those of you that plan on working in wildlife medicine, or research in general, it can be a little challenging and defeating. You know, you keep running into all of these challenges and it's frustrating. But by doing research and kind of giving these animals a voice is the only way that we're going to be able to remediate some of these issues. So I would encourage all of you to find areas of interest that you're passionate about and try and do your best to not only just publish in scientific literature, but get it out there to the general public in the general news media, social media, and reaching out to politicians and policymakers and people that manage different kinds of ecosystems, because just doing the research isn't enough. If you want to take it over the line and really help the environment or the population as a whole, you need to kind of make an impact on a broader scale. So I encourage you, for those of you that don't want to be vets, maybe go into policy. That is also a very effective way. But what we do is very reactive, and we need to figure out ways to be proactive and kind of start trying to fix these problems instead of just retrospectively study them and say like, oh, we shouldn't have done that. It's now a big problem, and then just leaving it at that. So I think that's all I have. I'm happy to stay on and answer more questions if anyone would like me to, but otherwise I really appreciate your time, and I'll get back to the other questions as quickly as possible.